Or my vision, dude.
You good? Okay, so we are trying, our title is Purification of Salmonella, Typhimurium Transcription Factors, Hill C and Hill D.
So we're working with this bacteria called Salmonella typhimurium, and it's a bacteria that causes mammalian gastroenteritis, and it pollutes food and water, and it kills over 100,000 people each year. Symptoms vary from diarrhea and abdominal cramps to septic infections, and can lead to death sometimes. The two transcription factors we're looking at are Hill C and Hill D, and they influence virulence and toxicity in this bacteria.
So the origins of research, at the Cool Lab at Dartmouth College, they had previously solved the crystal structure for TOX-T, which is an RSE superfamily transcription factor in Vibrio cholera, and it regulates the virulence cascade of Vibrio cholera. And when they solved the structure, they found a fatty acid bound to the ligand binding pocket of TOX-T, which in turn inhibits the DNA binding domain of the protein, which makes it ineffective and cuts off the toxicity. So when they looked at the structures, because Hill C and Hill D are also members of the RSE superfamily, they presumed that the structures of these two proteins are going to be relatively similar to TOX-T's in that it could possibly have a ligand binding pocket that could be inhibited by a fatty acid. So that's why we're looking at these two proteins.
So Hill C and Hill D, like we said, they regulate virulence and toxicity, but they don't do that directly. They regulate the primary toxicity factor called Hill A, and Hill A directly regulates the virulence and toxicity of salmonella. Hill A cannot be directly regulated because it's not, they found through analysis of the, it can't be because it's not structurally similar enough to TOX-T, so they don't want to deal with Hill A.
Can you repeat that?
Okay, so they did gene analysis of Hill A and Hill D and Hill C, and they found that Hill C and Hill D are most similar to TOX-T. So instead of working with Hill A, which may not have the ligand binding pocket, they did, the Dartmouth lab decided that it might be a better idea for us to work with Hill D and Hill C. So inhibiting Hill C and Hill D with fatty acid could shut down the virulence cascade, like TOX-T did in cholera.
So our ultimate goals for this year are to isolate and crystallize Hill C and Hill D. Our immediate goals are to first clone the genes into a PTXV1 expression vector, then transform the DNA into an expression cell line, which is PL21, then induce protein expression with IPTG, and then purify proteins through chitin binding columns and chromatography. Other overall project goals, which we probably won't get to this year, are growing crystals, which we might get to at the end, but these last two are out of our scope. They are to analyze the crystal structures at the Brookhaven National Laboratories and to ultimately solve the structures of the proteins, and we don't have the equipment or the money to do that.
So yeah, we just want to clarify, we wouldn't even be doing the analysis. We'd send the crystals off to Brookhaven and they do the analysis and they solve the structure, but we wouldn't be doing any of that.
So you're cloning?
Yes.
Potentially
Protein
growing
purification.
crystals.
Yeah, and then we're going to grow
crystallized.
crystals hopefully, and then send them to Brookhaven for
For
analysis.
structural analysis.
So our budget should be very little because most of the things that we do are also done in research class. So many of the things like Hill C and Hill D we're going to get from the Cool Lab. PTXV1 we already have from last year, so we don't really need to spend any money on that. Plasmid expression cells we have, but very little cost. Restocking plasmid and cell lines if we run out. Sequencing of our DNA after PCRing, and ordering primers for PCR and chitin beads. So it should be minimal cost. That's it. Questions?
So, can you go back to your big picture slide again, now that I know the plan? Tell me again exactly how this relates to the salmonella that you're talking about?
So, how Hill C and Hill D relate to something?
Yes, please.
So, salmonella, if you're infected with salmonella, you get sick. And the two transcription factors that will indirectly regulate the sickness, the virulence, and toxicity of Salmonella are Hilsi and Hildi. So if we're able to find the fatty acid that potentially inhibits Hilsi and Hildi, we would be able to potentially find some sort of treatment for salmonellosis.
What's the current treatment?
I think it's just, yeah, it's water, rest. It's, I guess you would be able to use antibiotics.
Nothing but a really concrete
Yeah.
temperature as of
Or if you don't have clean water, that's not a problem.
Yeah.
exactly, yeah.